August 4, 2023, Cambridge, Massachusetts. A researcher named Éadaoin Harney is sitting in front of a screen. On that screen are DNA sequences, extracted from 27 sets of human remains pulled out of the ground at an iron forge in Maryland, a forge that ran on enslaved labor from 1774 to 1850. She types a single query into a database containing the genetic profiles of 9 million living people. She presses enter. 41,799 names appear. Let that number sit for a second. 27 graves. 41,799 living relatives found in one afternoon. You've been told Black American DNA is unique, probably more than once. You've seen the videos, read the headlines, heard the claim repeated until it stopped feeling like a claim and started feeling like common knowledge. Here's the thing about common knowledge. It usually skips the part that actually matters. The word unique in genomics is not a compliment. It is a diagnosis, and a diagnosis implies a cause. What the Catoctin data showed wasn't just that these 27 individuals had living descendants. It showed that their ancestry profiles were anomalous, even within an already extraordinary population. Their genomic signatures traced back most strongly to the Wolof and Mandinka of Senegambia, and to the Congo of Angola and the Democratic Republic of Congo. But most enslaved people brought to the Chesapeake region came from Igbo and Yoruba populations in what is now Nigeria. These 27 were genetically different from the majority of people they were enslaved alongside. They were anomalous within an anomaly. 3 of those 27 individuals carried copies of the sickle cell gene variant. 4 carried a red blood cell enzyme deficiency called G6PD. These aren't incidental medical footnotes. Both variants cluster in malaria-endemic coastal zones of West Africa. Their bodies recorded, in molecular detail, exactly which part of that coastline they came from. No shipping manifest did that. No colonial record did that. The genome did. Stay with me, because this next part is the reason this video exists. If 27 graves in Maryland yielded 41,799 living matches, we are not talking about a historical curiosity. We are talking about a forensic event. And the genome is not just a record of who these people were, it is the only honest account of what was done to them. Every percentage point of that uniqueness means something. Every haplogroup frequency is a number, and numbers, unlike history, cannot be falsified. If you've ever actually wondered where you come from, not the family story, but the real biological answer, subscribe. Because what comes next goes deeper than most people are willing to go. The genomic record of Black Americans is not just distinctive, it is, in specific and measurable ways, the most precise forensic record of a crime that history tried to erase. And we have only just started reading it. Actually, before we go any further, I want to ask you something directly. Think about your own family. Is there one place, one origin, one branch, one gap in the story that you've always wondered about but never found a real answer to? Drop it in the comments. I mean that. Because by the end of this video, you're going to understand why that gap isn't an accident, and what your DNA might actually say about it. Silenced. That's the word that keeps appearing in the data when you look closely enough. Not missing, not lost. Silenced. In 2020, a team of researchers at 23andMe did something that sounds straightforward, but had never actually been done at this scale. They pulled up the Transatlantic Slave Trade Database, a historical record of 36,000 documented slaving voyages, and placed it next to genomic data from 50,000 living people across 12 countries. The question they were asking was simple: do the genetic patterns in living descendants match the population patterns in the shipping records? In several critical regions, they did not match at all. Stephen Micheletti led that study. His team found that Nigerian ancestry, specifically Igbo and Yoruba genetic markers, is dramatically overrepresented in Black Americans when you compare the genomic data to what the historical shipping logs would predict. At the same time, Senegambian ancestry is significantly underrepresented. And that gap matters enormously, because more than 1.8 million enslaved people were documented as coming from the Senegambia region. The ships recorded them. The DNA didn't keep them. The working hypothesis, and this is where it gets uncomfortable, is that Senegambian-descended enslaved people were disproportionately sent to malaria-endemic plantations in the American South, where they died at higher rates than populations with different disease resistance profiles. And when they died before having children, their genes died with them. The uniqueness of Black American DNA is not just shaped by who was taken from Africa, it is shaped by who survived the destination. And yet, the data revealed the exact opposite of what the standard history implies. The history says the slave trade was an act of transportation. The genome says it was also an act of selection. Not intentional genetic selection, but biological selection enforced by the deliberate placement of human beings in environments designed to work them to death. The chromosome doesn't care about the intention. It only records the outcome. Think about what that means for a second. The historical record was always going to be incomplete. Names erased, ethnicities destroyed, origins stripped away. That was by design. But the genome cannot be confiscated. It doesn't care whether a plantation owner recorded the ethnic origin of the person he owned. The DNA kept that information in a language no one could take away. The shipping manifests tried to reduce people to cargo. The genome kept a ledger. And that ledger shows something the ships were too efficient to record: who didn't survive the destination. We are only halfway through the list of what was erased. Because the story of which African peoples were functionally eliminated from the American gene pool doesn't end with malaria. It goes somewhere darker and more specific. And the data names a mechanism, not just a mortality rate. Elimination leaves a mark, not just in absence, in what replaces it. Charleston, South Carolina, 2022. Archaeologists and geneticists, working alongside the Gullah Society of Charleston, whose community history is directly tied to this site, are processing DNA extracted from 36 skeletal remains uncovered during construction near the city's historic port. The individuals are African descended, late 18th century. They were found a few hundred meters from what was, for most of the colonial period, the busiest slave trading port in North America. The team prepared 18 low-coverage genomes, and what those genomes showed immediately was not what anyone had predicted. Among those 36 individuals, buried in the same soil, at the same site, at roughly the same time, the researchers could not identify a single autosomal kin relationship. No siblings, no parent-child pairs, no cousins. In a burial population of 36 people, you would expect to find genetic relatives if the group had any natural community structure at all. There were none. These people did not come from the same families. They probably did not come from the same villages. They may not have shared a language. They were almost certainly strangers to each other. That is not an incidental observation. That is forensic evidence of a policy. The deliberate fragmentation of African family units, moving individuals away from genetic kin, deliberately mixing unrelated people from different ethnic backgrounds, was not an accident of the trade's logistics. It was operational. Prevent community, prevent shared language, prevent the foundation of organized resistance. And it left a specific mark in the genome. When you build a population from dozens of unrelated individuals, drawn from across dozens of different West African ethnic groups, you don't create a depleted gene pool. You create something geneticists call founder heterogeneity, a population assembled not from a single community, but from an extraordinarily broad cross-section of unrelated lines. The result is a gene pool with higher baseline genetic diversity than almost any other group of comparable size in the Western Hemisphere. The uniqueness is real. It was engineered. Not to benefit the people it was imposed on, but as a mechanism of control that had an unintended genomic consequence. The sickle cell carrier frequencies in the Catoctin remains confirm this. Those variants cluster in specific malaria-endemic West African coastal zones. They tell you not just that these individuals had West African ancestry, they tell you which part of the coast. Their bones remembered what no document was allowed to record. This isn't just ancient history. The genomic consequences of the slave trade are still being expressed in living people, in disease risk profiles, in carrier frequencies, in the specific chromosomal patterns that map directly onto what was done in the 17th, 18th, and 19th centuries. The past is not past. It is present tense in every genome. And the question that the Charleston data raises, and cannot answer, is whether there are other biological records still being expressed. Records not of ancestry, but of what the body learned to survive. Memory is the wrong word for what the genome carries. Memory implies consciousness. What the genome carries is a record, and this particular record is far older than anyone expected. Shum Laka, Cameroon, approximately 8000 BCE, a rock shelter overlooking a valley that will, thousands of years later, become the probable homeland of the Bantu language expansion, the population movement that will eventually seed most of sub-Saharan Africa with the ancestors of the people whose descendants will be loaded onto ships. In this rock shelter, 2 children are buried together, a boy and a girl. They will not have their genomes sequenced until 2020. When they are, the results force a complete revision of where the West African ancestry in Black American DNA actually begins. Dr. Reich Lipson and Dr. David Reich, both at Harvard Medical School, published those results in Nature. What the Shum Laka genome showed was a deep West African forager ancestry lineage, distinct from modern Bantu-speaking populations, distinct from Central African hunter-gatherers, something older than both, a genetic substrate that predates the agricultural revolution in Africa, predates the Bantu expansion, predates any civilization or political entity you have ever studied. And one of the 4 Shum Laka individuals, one of them, carried haplogroup A00 on his Y chromosome. Here's why that matters. A00 is the deepest rooting human Y chromosome lineage known to science. Its estimated divergence from all other human male lineages is over 340,000 years. For reference, anatomically modern Homo sapiens as a defined form emerged somewhere between 200,000 and 300,000 years ago. The most recent common male ancestor of the Shum Laka individual and every other man alive today may have predated our species as it currently exists. This discovery was made 5 years ago. It barely registered outside specialist circles, until now. The Middle Passage didn't interrupt an ancient lineage, it transported one. The West African genetic substrate that was loaded onto those ships in the 16th through 19th centuries wasn't simply a product of West African civilization. It carried, within it, one of the oldest continuously expressed genetic lines in the documented archaeological record of humanity. The standard narrative of Black American genetic uniqueness starts in 1619. Shum Laka pushes the clock back 340,000 years, and the genomic thread from that Cameroonian rock shelter to a living person in Baltimore in 2025 is not metaphorical. It is molecular, it is measurable, it is unbroken. And no one, not one mainstream genetics documentary, not one YouTube channel in this genre, has explained what that actually means for the people who carry it. What predates a civilization predates its erasure. But what happens when the erasure is so complete that the only surviving record exists in the diaspora, not in the homeland? The Jolof Empire, modern Senegal, 15th century CE. The Wolof people, organized into a functioning state with its own military, trade networks, and diplomatic relationships with Portuguese merchants arriving on the coast, become one of the first West African populations systematically targeted by Atlantic slave traders. By the mid-16th century, Wolof men and women are being transported west in significant numbers. 500 years later, Éadaoin Harney's algorithm finds Wolof mitochondrial signatures in the DNA of 27 Marylanders who died before the United States was a generation old. The Wolof didn't disappear from the record, they survived in it, in someone else's continent, in someone else's language, in someone else's history. The Catoctin study's finding of strong Senegambian genetic affiliation in those 27 individuals was unexpected for a specific reason. Most enslaved Chesapeake region workers derived from Igbo and Yoruba populations. That's what the broader historical and genomic record shows. The Catoctin group's Wolof and Mandinka profile suggests a specific early colonial era procurement pattern, likely tied to Senegambian trade networks before the Niger Delta routes came to dominate the trade. These 27 individuals are a genomic record of the earlier phase of the Atlantic trade, a phase that most people, even well-read people, have not spent much time with. What they found next changed something fundamental about the direction of this research. Because here's the thing about the Wolof. They're still there. The Mandinka are still there. Their languages, their traditions, their communities present in West Africa today. But the genomic map of Wolof and Mandinka populations within West Africa shows gaps, specific lineage frequencies that are lower than historical population sizes would predict. And in some cases, those missing frequencies appear at rates 3 and 4 times higher than in the current West African source populations in African American genomic databases. The diaspora is not just a product of Africa. It has become a repository of African genomic history that Africa can no longer fully access on its own. The research relationship is inverting. African Americans are no longer only the subjects of studies tracing African ancestry. Their genomes are becoming source material for reconstructing West African population histories that were disrupted by the very trade that created the diaspora. The laboratory has a new direction, and it points both ways. But there is one specific lineage, one Y chromosome line, that appears in this data at rates that no one has properly explained. A lineage so present in Black American DNA and so diminished in the modern West African population it came from that geneticists have started asking a question that sounds almost impossible. Is this a ghost? A lineage assumed demographically marginal in West Africa, alive in the diaspora at rates that suggest the diaspora didn't just carry it. The diaspora may be the reason it survived at all. Actually, I want to pause here for a second. In our last video on Japan's hidden genome, we found that the oldest genetic lineage in the Japanese archipelago was nearly erased by a later population wave, and that erasure was not accidental. It was the way dominant populations write history, on top of the people who were already there. If you watched that one, I want to know what you think. Is the suppression of ancient genetic identity a deliberate policy, or just the natural consequence of who ends up in charge of the story? Drop your answer in the comments, because the answer to that question changes everything about what I'm about to show you. A ghost is what remains when the body is gone. But what if the body was never gone in the first place, and the ghost story was never the right explanation? There is a competing argument. It has peer-reviewed support. Several serious researchers at respected institutions have made versions of it, and I think you deserve to hear it fully. Not the weakened version, but the strongest possible case, before we go any further. The argument is this: Black American genetic uniqueness is not primarily a product of the transatlantic slave trade. It is a product of West Africa's pre-existing genetic architecture. An architecture so deep, so old, and so complex that any population drawn from it would appear uniquely diverse when compared to non-African populations. Under this reading, the slave trade didn't create the genetic distinctiveness. It simply relocated it. This is not a fringe position. Pontus Skoglund's team at the Max Planck Institute for the Science of Human History published work in Cell in 2017 demonstrating that genetic diversity in sub-Saharan Africa dwarfs the genetic diversity of all non-African populations combined. Africa contains more genetic variation within a 200-kilometer radius than exists across the entire Eurasian supercontinent. If that's true, and it is, then any forced migration of West Africans would automatically produce a genetically diverse diaspora population, regardless of any demographic bottleneck at the destination. The bottleneck theory in this reading is redundant. The uniqueness was always going to be there. This isn't just ancient history, this is in your DNA right now, and the question of what put it there matters enormously. Here is why the counter-argument fails. One number. One specific, non-negotiable number that the counter-thesis cannot absorb. The Micheletti study didn't just track ancestry, it tracked the ratio of African female to African male genomic contribution across 12 countries in the Americas. And in Central America, that ratio reaches 13:1. In parts of South America, 17:1. 17:1. No natural migration event produces a 17:1 sex ratio in genomic contribution. Not famine, not disease, not any demographic process that operates on a population without external direction. Pre-existing West African diversity, however extraordinary, does not explain a 17:1 female-to-male genetic contribution in the diaspora. Because that ratio is not a feature of West African genetics. It is a feature of what happened to African men after the ships docked. The bottleneck is not at the origin, it is at the destination, and it is American. The counter-thesis actually strengthens the primary argument when you apply this number to it. Because if the pre-existing West African genetic diversity is real, and it is, then what was suppressed in the Americas was not random variation. It was the male side of one of the most genetically diverse human populations on Earth. The cost compounds. The loss is larger than it would have been if the population had been less diverse to begin with. And here is the part that most people are not prepared for. The biological consequences of that suppression did not end at emancipation. A study published in PLOS Genetics in 2016 tracked the genomic footprint of the Great Migration, the movement of approximately 6 million Black Americans from the South to northern and western cities between 1910 and 1970. The sex-biased genetic patterns from the slave era, the specific haplogroup distribution asymmetries created during the 17th and 18th centuries, were still visible in regional American genomic data as recently as 1970. The biological war did not end when the legal one did. A war leaves casualties. Genetics counts them. Somewhere on the Atlantic coast of Latin America, approximately 1700, a ship discharges its cargo. The manifest records over 300 enslaved persons. More than half of them are male. Within one generation, the genomic record of those men will have almost entirely vanished from the local gene pool. Not because they died on the ocean, because of what was done to them after they arrived. The Micheletti study is where this becomes a verdict rather than a theory. 50,000 people, 12 countries, the largest genomic study of African American and Afro-descendant populations ever conducted at the time of its publication. And what the data showed, across every country, across every time period, was the same pattern. African women contributed to the gene pool at dramatically higher rates than African men, despite men comprising over 60% of the enslaved population transported to the Americas. The study's language is scientific, consistent with a history in which interracial mating primarily happened between mothers with African ancestry and fathers with European ancestry. The translation is not scientific, and I'm going to say it plainly because the data demands it. The 17-to-1 ratio is not a statistic about women. It is a statistic about men. It is a precise genomic measurement of how completely the reproductive future of African men was suppressed, not by death alone, but by a system architecturally designed to prevent their biological legacy from surviving. The European male lineage replaced the African male lineage in the gene pool, not through natural population dynamics, through a structure of power that controlled who could have children, who could maintain family units, and whose genetic line would continue. Everything I just told you? Hold on to it, because it's about to mean something completely different. Here is the detonation, the single finding that the entire preceding hour of evidence has been building toward, and that changes what the word unique means permanently. Despite more than 12.5 million Africans crossing the Atlantic, the Y chromosome lineages of African men are so underrepresented in Black American DNA that geneticists can now prove, in base pairs, in haplogroup frequencies, in population-level data across a dozen countries, that the males of the Middle Passage were systematically prevented from reproducing, while their female counterparts contributed up to 17 times more to the surviving gene pool. This ratio has no parallel in any other documented migration event in human history. The uniqueness of Black American DNA is not only a record of who survived, it is a record of who was not allowed to. The women's names were not recorded, their ethnic origins were not recorded, their children's names were not recorded, but the mitochondria kept the accounting. Across 12 countries, across 300 years, in every population studied, the female mitochondrial signal strong, the corresponding paternal Y chromosome signal from the same ethnic groups fractional. The biological record was never destroyed, it was just waiting for someone to read it. Every Black American man watching this carries a Y chromosome that passed through that filter. That is not heritage as most people use the word. That is selection. And the conditions of that selection were not natural. The genomic record is specific enough now, region by region, time period by time period, that when it is cross-referenced against historical ownership records, it does not only describe institutions, it is beginning to describe people. Not systems, not eras, people. People. That is where this story returns, always. A federal genetics laboratory in Cambridge, 27 sets of remains, 9 million living profiles in the database, and 41,799 matches returned in the time it takes to make a cup of coffee. The system built to erase those 27 people, to strip their names, their ethnic origins, their family connections, their languages, failed. Not dramatically, quietly. in a spreadsheet, on a Tuesday afternoon. Because the genome is not a document, it cannot be confiscated, it cannot be altered by whoever holds legal title to a person. It is a molecular record that keeps accumulating precision with every decade of scientific advancement, regardless of what anyone intended 250 years ago. The Catoctin Furnace Ancient DNA Study, published in Science in August 2023, represents something genuinely new in the history of this field. Not just in what it found, but in how it was done. The research was conducted in direct partnership with the Catoctin Furnace Historical Society and with descendants of the enslaved community who had maintained stewardship of this history for generations. The 41,799 living matches were not simply published in a journal. The researchers worked to contact descendants, to return the story to the people it belonged to. That process is not complete, it is ongoing. And the Catoctin study is not an isolated achievement, it is the leading edge of a methodology. The same ancient DNA approach, pairing skeletal remains from enslaved burial sites with contemporary genomic databases, is now being applied at sites across Virginia, South Carolina, Louisiana, and Maryland. Each site, when sequenced, yields not just ancestry data, but a specific genomic profile that can be matched to living descendants. The machinery of reconnection is being built in real time. There is something else in the data that has received almost no public attention. Several of the Catoctin individuals carried genomic profiles with affinity to populations in present-day Senegambia, specifically populations that have seen significant demographic disruption since the 18th century due to internal African migrations and colonial-era border drawing. In some cases, the genomic profile of a person buried in Maryland in 1800 is a more complete representation of what a specific West African ethnic population looked like before that disruption than anything the modern descendants of that population have access to. The diaspora is carrying genomic history on behalf of Africa, not as a metaphor, as a literal scientific reality, which brings us to the question the research is not yet ready to answer, the question that the field knows is coming, that individual researchers are already asking in conference rooms and in the margins of papers, but that has not yet been stated in public with the weight it deserves. If Black American ancient DNA can be used to reconstruct West African pre-colonial population histories that no longer exist intact on the continent, what else is in there? What other histories are being preserved in the diaspora that have been lost everywhere else? We do not know yet. The sequencing is still running. The database is still growing. The 41,799 names were found last year. This time next year, the number will be different, larger. The accounting is still happening. There is one more thing, the thing I almost didn't include, because it sits at the edge of what the science has confirmed, and I will tell you exactly where that edge is. Among the genomic patterns that researchers have flagged in studies of African American ancestry, there is a class of variants that appear at statistically distinct frequencies in populations descended from high mortality historical events, stress response loci, immune regulatory markers, variants tied to the HPA axis, the body's core cortisol and stress response system. Several of these appear at measurable frequency differences when African American genomic profiles are compared to West African reference populations from groups not affected by the slave trade. I want to be precise about what this means and what it does not mean. The field has not reached consensus on whether these frequency differences represent selection pressure imposed by the specific conditions of enslavement, by malnutrition, by brutal labor, by the documented psychological conditions of that system, or whether they reflect pre-existing variation that was amplified by the demographic bottleneck. The research is ongoing. The debate is real. I will not present a preliminary finding as a settled fact. What I will tell you is what the researchers who have flagged these patterns are asking. They are asking whether the specific conditions of the Middle Passage and American plantation slavery, conditions documented in extraordinary biological and historical detail, left a measurable mark not just in ancestry frequencies, but in the regulatory variants that govern how the body responds to threat. They are asking whether some of what is being expressed in living African American bodies is not just the record of where their ancestors came from, but the biological record of what their ancestors endured. That question is not resolved. It may not be resolved in our lifetimes, but it is being asked by serious researchers at serious institutions with serious data. And the fact that it is being asked, the fact that it is now scientifically askable, is itself a product of the same genomic revolution that found 41,799 names in a Tuesday afternoon query. If you carry West African ancestry, if any part of your genetic line runs through the events this video has described, there is a non-trivial probability that you carry a variant shaped by one of the most consequential demographic events in recorded human history. Not shaped by heroism, not shaped by survival in the romantic sense, shaped by a specific, documented series of choices made by other people about what your ancestors were allowed to be. The genome didn't ask permission to keep that record, it just kept it. And here, before the last thing I need to say, if this kind of forensic genetic history is the work you want to keep following, subscribe and hit the bell, because the next episode goes somewhere you have not been before. The question the science leaves open, the one that no haplogroup frequency, no ancient DNA study, no population genomics database can answer, is what we do with a record this precise. The legal systems of most countries were not built to process genomic evidence of historical crimes. The cultural and political frameworks for reckoning with data this specific do not yet exist. The science has outpaced the institutions. 27 people were buried under the ground at Catoctin Furnace. Their names were not recorded. Their ethnicities were not recorded. Their children were not recorded. The system that put them in the ground was confident it had erased them completely. It was wrong. By 41,799 people and counting. The genome is the only witness that was never silenced. And it has been waiting in the dark, in the molecular architecture of millions of living people, for someone to finally ask it what it saw.